Hi everyone, my name is Maddie Wood, and today I'm going to talk a little bit about biotechnology and how CRISPR is being used to treat genetic diseases such as sickle cell. Genetic diseases have impacted living organisms since DNA was created, and these diseases are caused by mutations in DNA that can impact the development and function of these living beings. However, in an age of rapidly developing technologies, the field of biotechnology has given birth to genetic engineers that can fix these mutated genes using technologies like CRISPR-Cas9. Anyone's DNA is at risk of mutations due to genes they inherit from their parents and external factors such as diet, chemical exposure, and drug and alcohol use. So it's important to be aware of the new genetic treatment options available for all these diseases. I'm currently a biology major at Cal Poly, and I'm studying microbiology and genetics. And the topic of genetic engineering, especially related to these genetic diseases, is especially fascinating to me. Biotechnologies like CRISPR have the potential to cure genetic diseases such as sickle cell, improving and possibly even saving thousands of lives. Sickle cell disease is a prime example of how CRISPR has been used to treat genetic diseases, and investigating what sickle cell is, what causes it, and how to fix it can give better insight into how these biotechnologies work. So let's look a little closer at the sickle cell example. According to a 2022 article published by the FDA, sickle cell gets its name from its shape. Normal red blood cells, as you can see on the right, are nice and plump. They're large and round, and they can hold a lot of oxygen, which is the function of a red blood cell, is to carry oxygen throughout the body. However, sickle cell is crescent-shaped and very rigid, and this prevents it from carrying a lot of oxygen and also impacts its ability to flow through blood vessels, so it often gets clogged. Decreased oxygen levels in the blood can cause dizziness, weakness, fatigue, and other issues with bone density. And severe cases of sickle cell result in needing lifelong blood transfusions of healthy hemoglobin. According to a study conducted by the CDC in 2023, sickle cell is very common among those who have ancestors from Africa, Spanish-speaking countries in the Western Hemisphere, Saudi Arabia, India, and Mediterranean countries, and it is also pretty prevalent in the US. Although exact numbers are not known, the CDC estimates that as of 2023, sickle cell disease impacts around 100,000 out of the 340 million Americans, which is about 0.03%. Worldwide, around 250 million people have the sickle cell gene. This means that sickle cell disease could be expressed. However, they could also just be carriers and not be impacted at all. You may be wondering, what exactly is a genetic disease and what causes it? Genetic diseases are caused by errors in genetic material, and this could be caused by mutation of DNA through a replication process in an already developed individual, or it could be genetically inherited and incorporated into an embryo's DNA, which would impact its development. There are three types of genetic diseases. On the top left, you can see the chromosomal disease, and this is when there is an addition or subtraction of chromosomes, of entire chromosomes. On the very top left, you can see a normal set of chromosomes, and in the other three examples, you can see an addition or a subtraction, and this would impact how the individual develops, if certain parts of an individual are missing, things like that. Another type of genetic disease is multifactorial, and this is when a mutation is caused by a combination of genetics and external factors. The example we can see in this infograph, it's obesity, there's smoking, hypertension. This is stuff like diet, drug use. So these risk factors and these external factors can impact how things function within the body and can then impact the genes that within a person. Finally, monogenic is when one singular gene is impacted. And in this example, the orange would represent the mutated gene, and you can see how it is passed on from parent to offspring, but it is only a singular gene that is changed. Sickle cell is a monogenic disease. So that means only one gene is impacted, and the mutation in DNA that results in sickle cell is a result of a single base pair. You can see on the left, the healthy hemoglobin has CTC, and on the right, it goes CAC. So the difference between the T and the A will encode for different proteins, which will impact the overall structure of the final hemoglobin. Sickle cell is also an autosomal recessive trait. This means that only a person with two recessive alleles for this trait will be impacted by the disease. So many people will live their lives carrying only one recessive sickle cell gene and one dominant healthy gene without any health issues. As you can see here, a parent if two parents that have the gene were to cross and have children, there's a 25% chance, so 1 out of 4, that their children will have sickle cell disease. Is the bottom right is the 2 small s's. The top left, this is a child that does not have the gene and it is completely healthy. And the 2 corners, there's a 50% chance that the child would be healthy, however, they would carry the sickle cell trait. However, scientists have come up with ways to combat these genetic diseases. CRISPR is a gene editing tool that can be thought of somewhat as a genomic scissors. The Cas9 protein is used to cut DNA, where scientists can add or remove base pairs or large segments of the DNA. This technology is allowing scientists and doctors to edit and modify and correct mutated parts of DNA. Prior to genetic treatment, the only FDA-approved treatment for sickle cell was bone marrow or stem cell transplants. However, around 15 years ago, scientists discovered a gene called BCL11A, and this controls the amount of fetal hemoglobin that is released throughout the body. The fetal hemoglobin is able to control the shape of blood cells, and more fetal hemoglobin means more healthy red blood cells. However, it's not usually released past the completion of fetal development, so the scientists needed to figure out a way to turn this production back on to increase the amount of healthy blood cells in those suffering from sickle cell disease. Using CRISPR, BCL11A was turned off, which resulted in an increase in fetal hemoglobin expression. Due to the monogenic nature of the disease, this treatment was relatively simple as genetic treatments go, and only 1 gene had to be manipulated to get rid of these sickled cells. This is why CRISPR technology treatment for sickle cell disease was the first genetic alteration treatment to be approved by the FDA in 2021. Biotechnologies like CRISPR have the potential to cure genetic diseases like sickle cell disease, improving and possibly even saving thousands of lives. CRISPR has the potential to treat a variety of genetic diseases, just like cystic fibrosis or Huntington's, and understanding how scientists have successfully used CRISPR on diseases like sickle cell can help create treatments for other diseases. This can start with understanding what sickle cell is, what causes it, and how it is fixed. DNA is the basis of all living things, and it's important to know how to keep this sacred code safe. However, when it's inevitably mutated, it's also important to know how to fix it. Utilizing biotechnologies like CRISPR can completely remove these diseases that have plagued species for hundreds of thousands of years.